Informatics, professor of Department of Pathology, Cherish Endowed Chair at Stony Brook, and I had the privilege to work with Joel at NASA Goddard, where we was in the institute, which was called Center of Excellence Space and Information Scientist. Joel has MD and PhD from Duke University, and over the years built number of national departments in bioinformatics and conducted the world research, and he's a world-renowned scientist in computer science and in pathology, artificial intelligence, and many others. Without further ado, Joel, welcome to the University of Miami, and great to reconnect.
Thank you for that kind introduction, and it's really wonderful to be here. Beautiful campus, great people. I really had a great day, and thank you. So I'm going to give a overview, primarily of much of the work, some of the work we're doing in the area of digital pathology and the development of methods to segment, classify images, predict outcome, response to treatment, and contextualize this as to where this sort of thing fits into medical research and current state of technology, as well as some of our own work. I'm also going to talk about clinical informatics and digital health briefly, because what the main part of my talk is digital health as well. But I know that there's a lot of interest both here and other places in digital health. And, Data Science Institute has a lot of interest in this. So I thought that it might be worthwhile to first talk about a number of the projects we're doing in digital health and how they relate to computer science. Because I think in much of the world thinks to people in the institute here is that digital health is both important in and of itself, but also can act as drivers for interesting computer science research. So I thought that I would first give sort of an overview of that, at least from sort of my own local perspective, at least work that I have done. So multimodal methods for predicting outcome in response to treatment. What I mean by that is gathering imaging data, pathology data, molecular data, sensing data you need to get your hands on, and using this to predict effectiveness of medical treatment and to assess what is going to go on. So this is obviously an incredibly general statement. I'll be talking about much more specific work that we do in digital pathology, but at some level, a good fraction of work in digital health, whether it's response planning, predicting response to a surgical intervention, predicting response to radiation treatment, predicting response to a medication, or whatever it is. This is very broad. Agile methods for query visualization analysis of multimodal data sets. Again, there are a large number of projects that involve query and analysis, including actually the talk earlier today. People were at it that deal with this sort of issue. This is very popular. Real-time active super resolution clinical data acquisition. This is a cyber physical system. Dynamic data-driven application system sorts of things.
Trouble hearing when you're not listening.
Can you hear me?
So I'll go through this part, but suffice it to say that digital health and digital health problems are great drivers for computer science research. And I list a number of different areas in digital health and computer science here. I won't go through each of them in the interest of time. But Suffice it to say that actually it is possible, like our group and many other groups have managed to essentially abstract areas or themes in digital health, and then sort of ask, well, what are some of the basic computer science problems that are behind these areas? Taking care to try to generalize, to say, okay, our goal is not just to solve the specific problem in digital health, but to find sort of broader principles that would address a broader set of applications. So in my view, preferably including some applications that are actually not health applications. That tends to be a good way to demonstrate that the principles are general. So these are some examples of work that my group and I have done over the years, motivated by digital health and areas of computing and computer vision and system software. And areas of digital health are varied and many. And our group at Stony Brook consists of a number of PhD faculty, a number of physicians who are either have been or are undergoing subspecialty training in an area called Clinical Informatics. These are people who finished their residency and have spent a 2-year apprenticeships working with us to solve practical problems and more theoretical ones with the non-systematic. So these are people who are not in medical school, they did residencies and internal medicine, like pathology, and so it's a very it's an interesting team. Okay. So it's an interesting team that work together on these sorts of things. And then I'm going to give you this is sort of a laundry list of some of the digital health projects that at various institutions, including Ohio State and Hopkins and Emory, that our group has been involved in. I won't try to read these, but it's a very varied number of things. So forth. So digital health, in my view, has both a research component where you want to be relatively narrow, go deep, and then it often has much more of a service component where you actually have a much broad, you're basically responsive to problems in your health system. And so, for instance, one new project that we have involves capturing model patient flows to the emergency department and health system. People will spend patients in our places, other places, spend more time than months held down the emergency department facility in the hospital. So the chief medical officer contact us and said, "Can you help us with this?" So we're using the but the object is health constant. So and then often these projects do projects that are much more researching. So that's sort of and we've given much of our attention to projects that involve imaging and digital pathology, but often with great knowledge of outcome. And that's sort of the space of projects we're working on group and radiation oncology. So I also want to give a shout out to our education programs. And these were mostly organized by Mary Saltz. And there's a PhD program, master's program, boot camps, various training sessions for medical staff, sessions involved underserved populations, high school students, we can enter in various ways. So it's quite a, but that's a whole other talk. And then also we're involved in writing book chapters for education. Okay, so now I'm going to move on to the future presentation and talk about digital pathology, speed learning, and things having to do with developing essentially new ways of predicting what amounts to outcome response to treatment, disease classification. So traditionally, pathology has been around to discipline for 150 plus years, and there are a very large number of human generated classifications in pathology, which have developed to a large degree because pathologists and oncologists, clinicians of various sorts, have found them to be predictive. And over the last 50 to 100 years, there's been an increased tendency to formalize this, develop systematic studies to document that if one has a patient with brain tumor type X, you can expect Y to happen on average. So these basically, you look through the microscope and see these patterns, and they have names. They've been given names by people. There are tens of thousands of names, and pathologists over their 3-year anatomic pathology residency learn how to associate names with patterns of it. And that's kind of what it amounts to. And there's a paper that came out a few years ago by Levinson on the West Coast, UC Davis, where he actually trained pigeons to do pathology tests. For pigeons, don't have 10,000 categories. But and as I'll talk about, you can train neural networks to do many of these things as well. But here's the key point. The point is that these names are cultural. These names come from people who happen to have either engaging or strong or whatever sort of personality. I will call it this. And they're usually packages of different attributes that a human kind of put into a name, and then they teach each other about the name. And the name is predictive, but it's not necessarily the most predictive thing. It just happens to be what was culturally sort of promulgated by a combination of medical science, politics, who is from Hopkins or whatever it happens to be. So in the last few years, it's become possible to do things totally different, which basically involves taking a whole slide image. So basically a slide, an image of a glass slide consists of something in the order of 100,000 by 100,000 pixels, so they're megapixel images, and essentially systematically, electronically circling and labeling every cell, every duct, every crypt, every little structure. So you have, essentially what is analogous to a Lego set. So you break the thing up into zillions of little pieces. And then you ask, how do I reassemble these little pieces to get new methods for predicting treatment response and what's going to happen? So it's really a fundamentally different way of dealing with this. It's a little bit like if you've ever watched Mad Men and you looked at advertising in the 60s and 70s, and it was all inspiration. And a lot of it was really smart. And you look at Google and you look at the current approach to advertising, where you have huge amounts of data, lots of feedback. You still need inspiration, but in terms of data science, this is really data science. Huge amounts of data in these little tinker toys. So we and other folks have been developing a lot of methods, but what I'm going to talk about mostly is a set of methods that have become relatively mature, involving characterization of immune cells in tumor. And immune cells in tumor, as the Miami Radon knows very well, are incredibly important. So one of the things that they actually showed me in prostate cancer, and to be honest, is that if you have a patient, so one of their patients actually, I guess, came in with prostate cancer, a part of the prostate. And then the next year, they were followed, that cancer regressed or even went away. And there was another place of the prostate. So the thing didn't jump around. What was happening almost certainly is the patient's immune system was fighting the cancer, but it would gain a temporary victory, beat the thing to death, and then it might recur someplace else. Most men die with prostate cancer rather than of it. Most breast cancer is not fatal. Most people, if they get hit by a truck, if you do an autopsy, would have many little foci of cancer. So it's not like, oh my God, they have cancer, we have to treat immediately. There's a very dynamic interaction between the immune system. And now there are lots of treatments that explicitly involve helping the immune system fight a cancer. And so understanding when those treatments might be useful and deciding which one, how to combine with other treatments such as radiation therapy and standard chemotherapy and targeted therapy is very important. So suffice it to say, the issue of finding tumor infiltrating lymphocytes. You have a tumor. We have here is a picture, got yellow and red. So up here you've got the upper left-hand side, the thumbnail or the page to the image. The real image is huge, but this is very, very low power. What you have here is the result of a deep learning prediction, which tells you where the tumor is, which is yellow, and where the immune cells are, which is red. And you get very characteristic patterns of these things. You can also zoom in and look at individual cells, although getting the ability to resolve a cellular resolution reliably takes more work. So these are a set of algorithms that actually our group really developed some of the first ones in 2017. They're now pretty routine, and we use them now in various collaborations because they're really pretty solid. So I'm going to talk about some of how they're used in the context, and then I'm going to talk about some sort of both why you want to use such things in the research for new algorithms, new methods. So now that these things are pretty solid, then we're now in a position to work with clinical research groups and consortia to look at the relationship between having lymphocytes, having immune cells and tumors, and the patterns of immune cells and tumors, and what happens to patients. So we work with a cancer cooperative group called ECOG Akron, building this into some trials. We have a consortium funded by the National Cancer Institute, consisting of cancer centers with something called SEER sites, which involve NCI-funded curation of data. And the proposal we said, what the call for proposals was to add new information to SEER data. SEER data comes from essentially pathology reports, it's curated, plus some other data. So we're adding pathomic data, we're adding sort of tumor infiltrating lymphocyte data, nuclear segmentation data to that. And then in particular, the focus studies we're doing involve real world studies of the impact of tumor infiltrating lymphocytes on outcome, different types of cancer, faceted by different clinical variables. We got into this in, as I said, in 2017, 2018, the Pan Cancer Atlas. So the TCGA had a variety of tumor groups, and there was one that involved spanning multiple cancers. So they were looking at molecular characterization of across cancer, what pathways were relevant. So part of this involved looking at immune tumor interaction. So they were interested in working with us on computer vision. So basically complementing their molecular data with image analysis. And so there's a whole validation process that we went through to make sure we were getting the right answer that I won't get into. But what I will talk about is that once you get into this sort of spatial description of immune cells, and probably many of you have heard about spatial transcriptomics there's an increasingly large set of platforms, and I'll talk a little bit about some of our work there that involves, multiplex spatial RNA proteins, mass spec and the like. But one way or the other, you have to describe spatial patterns, which leads to a whole spatial statistics sort of thing. So in the TCGA Pan Cancer Atlas project, we worked with a wonderful spatial statistics person who borrowed a bunch of methods from earth sciences and pathology to characterize patterns of clusters of immune cells. And the data we generated from this is all public. And so there's molecular data involving both the tumor and the immune system, and different types of cells at the whole slide level. And then there's percentage of TILs, and then there were various parameters to describe the patterns of clustering. And then given that, one can look at the pattern of the impact and outcome and relationship to other molecular data. So that was a very rich data set. It's still, we continue to go back to what actually other groups do as well. But we decided after we published this in Nature Reports and Nature Radiology with this huge consortium, one of the things we wanted to do is to take a deeper dive and look at the impact of patterns of TILs in particular diseases where the essentially clinical research story is a little clearer. So we have this wonderful collaboration with Melissa Troster and the group at UNC, as well as Etta Pisano at American College of Radiology, where what we've done so far is to characterize patterns of TILs in a slightly more human interpretive form than those parameterized distribution functions. So I won't go into all of these things, but for instance, you've got a pattern of forests, you have lots of TILs in certain areas, or a pattern of deserts where you've got a lot of tumor, no TILs, or you have peritumoral where there are TILs surrounding the tumor. So you have various different, patterns, and there are obviously lots of different ways of describing patterns. This is kind of a Lego thing and done. So there are lots of ways of doing it, but then given those patterns, given percent TILs, what we've done is to break down by different TCGA or different breast cancer subclass and look at the impact percentage of TILs on survival. And this involves both the TCGA data set and the Carolina Breast Consortium. An incidental point, TCGA is a wonderful source of data. In many cases, though, you really want to have validation data at the minimum from a separate data set, because much of what we know about now comes from TCGA. So there's a potential for a bit of overtraining. But then actually one of the things that we did was to do hazard ratio risk factor analysis. So this is a little bit like you probably run into in heart disease, lipid levels, things like that. How many risk factors do you have? So we basically had a number of different risk factors involved, patterns of TILs, and then we calculate hazard ratios. And suffice it to say, it ends up being that having several risk factors seems to be important. So this is one of the things that we're doing in terms of validating this. One is with ECOG ACRIN data, and the other is with real world data from our SEER collection of SEER sites. So, so one of the things that really is that you have one thread of work, which is to take algorithms that are validated to give you much better precision, and then painstakingly go through multiple datasets. And I should say, we're now in the past the beginning. It's end of the beginning. We're now in the middle, working with the Miami, because we have the data, have all the agreements. And the notion is to do similar analysis with prostate and to look at their active surveillance groups. They're watching prostate cancer. Some of them progress and some of them don't, and then try to do the same sort of predictive kind of work. So we also generate more nuanced maps that I won't get into, involving different tumor subtypes, and that also interplays with TILs in various ways. We also have a project on inflammatory bowel disease. So I'm going to fast forward here. So I've given you so far a description of how we developed an algorithm, which actually, believe it or not, was 2017 publishable in some nice places. Now it's totally routine. And over the last few years, now working with collaborators to use that to explore ways of predicting what I've described as response, but in both the Carolina breast study and also we'll do this in Miami, certainly breast study response to treatment, because as you get more and more time goes by and more patients are treated with new therapies, it's a very telling story as okay, we're able to characterize the new tumor distribution, and you can look at the therapies which relate to that. But then another thing that you really want to be able to do is to continue to improve the methods. And we have lots of great collaborations between both the biomedical and computer science, looking at various different issues. So one issue that we've run into the program is we want to be able to do is at scale. What I mean by at scale is you've got tens of thousands, hundred thousands, millions of standard gigapixel images. Each one of these images, if it's from the precision, is going to have like 1 million or 2 million cells. So if you want to be able to detect, label, segment, classify what amounts to trillions of cells, then you need algorithms that are pretty reliable. I mean, because the old profit algorithms from 10 years ago, we found that it would take we had a bunch of undergrads who did error correction, and it would take a couple of hours per whole story image to fix up the results. These things, millions of cells. So better algorithms actually are very important for scaling. And so one theme in our group is going to apply pathology to deep learning algorithms. So deep learning algorithms, you do 80% of loss functions, which try to optimize for various things. And one of the things you can have is geometrical and topological considerations. So in this particular algorithm, what's used is something called the K function. So there's the Ripley's K function. This is a generalized version where you basically characterize local spatial statistics and characters of what are the distribution of cells. And then that becomes part of what you're trying to optimize. In other words, if you say, look, we can detect, so we have training data that says we have this distribution of cells. And then when you're doing your optimization, say, okay, this is what we're expecting, and that they should have a certain spatial characterization. So this is, I sort of call this cells of a feather flock together. And so typically you often do have certain sorts of cells that are neighbors, but they're not all that sort of cell. They just increase statistical tendency to have them. And this seems to be very, so we published this at a couple of computer vision conferences, and we're using this now for some of our practical stuff. There's also, as part of the gradient descent clustering process. So, and this comes up in actually a number of algorithms these days. There's an algorithm called plan from which we're using as well. And he also has sort of basically in the middle of the loss function, in the middle of your optimizations. And by the way, one of the things I want to have is the two classes be separate mechanisms. Right, it makes sense. It's like if there's other classes, basically go to the middle and they're all that. Separate these things. So this is actually sort of I think a nice example of a kind of, in some sense, an engineered deep learning algorithm. Like back in the day with radiomics, there were the individual treat parameters. But now, what one of the things you can do is say, okay, I'm going to engineer a neural network pipeline, which is going to have a bunch of criteria stuck in the optimization, like keep the different classes separate and make sure that the spatial properties of locality are a certain way. So, but other than that, just do the usual thing. So this is moving along in digital pathology and obviously how these things, these same considerations that take place in pathology. Some folks see topological separation of organs in the radiologic image. So then we do actually tackle the issue of multiplex spatial. This is spatial proteomics, and this is at the whole slide imaging level, and involves pancreatic ductal carcinoma. And so you hear a lot about spatial omics. Hear about it at all. But one of the things that is not often recognized is most of these platforms and most of these studies do work on little ROIs. And in doing work on whole slide images, it's a whole different because it's a huge amount of data and validations that typically deal. And there's actually a Hopkins group led by Helen Soleil and a bunch of folks who do stuff there. We're doing multi-spectral immunofluorescence, sort of similar scenario. And I've used this we're doing whole slide images. Nobody else has whole slide images, and we are. So there are a few of us here, and this is multiplex IHC. So this is an example of one of the sorts of problems we deal with. So basically, you've got a tumor marker, and this is sort of a mechanism stuff. So if you've got the bad sort of, well, all types of pancreatic carcinoma, but they're the bad ones is even worse ones. And so the even worse ones are basal, the bad ones are classical, and a bunch of folks in our general research collaboration friend group deal with this both in molecular and morphological terms. So as well as mechanistic. So here you've got basically a tumor. So basically the brown and the brown ones are slated with a marker that basically tells you you've got even worse one. And the teal ones are not as bad. So a whole patient gets classified as really bad or worse. But in practice, actually, when you look at the whole slide, it's heterogeneous. So then the question that our collaborator wanted to ask was, how does the even worse one the pattern of different types of immune cells close by? And the challenge there was, one is you have this child's colored paper that you've got to dissolve the cells. So that took the graduates to a couple of graduate students a year or two to get a good solution, and we are using it and published it. And then the other is to do the spatial analysis involving the locality of these irregular boundaries. So this is an example of the result where you're looking at CD8s, and this red is essentially concentration cells that are close, CD8 cells that are close to even worse, and then we are CD8 cells that are close to the teal area. But remember, you can have different ratios of teal versus brown. So you're not interested in the absolute amount. You want the amounts that are close by, sort of a spatial analysis. And then, fortunately for our collaborator, this did seem to bear out his hypotheses that basically you have more CD8 cells close to the not as bad part. So the bad part is CD8 cells less likely to be close by. So I'll now move into, I don't know, how long should I go? Because I don't want to put everybody to sleep. So we're not. Okay. All right. So okay. So I've given you an example. Of course, be happy to. Yeah. No.
I was just asking, when you talk about spatial analysis, do you ever because I know whole slide images are much more like accessible as data than like some sort of video screen. I like the fact that there's like, if you have like a flat slice of like out in,
Yeah.
you know what I'm
Well,
saying?
no, I totally know what you're saying. And the essence, yes. Written a number of papers about it. We think this really very very labor intensive study, actually the whole state still there, involving looking at mouse placenta. And our collaborators have been that RV kind of had some impact on the funding problem, whatever. And so it involved like 1,000 slices of mouse placenta, and then we have to do this 3D reconstruction of the thing. And actually, Raghu Mashkarashta, he was a faculty at Ohio State. Maybe he was. Yeah, but anyway, so he played it. Yeah, so and then since then we've done it to a degree. Not as okay. So here's the issue with 3D reconstruction. Okay, the right way to do 3D reconstruction is probably not to do 3D reconstruction, but okay, and this is something I'd love to do. Is basically to do super resolution reconstruction of something like micro CT of the block, and then use a couple of slices at high power to sort of drive this, right? Because that way you get a lot more tissue. That way you don't have to reconstruct it because it's 3D already. You go knocking your at MSKCC was doing some nice work, kind of a little bit along those lines. But and so I think something like that is the right thing to do. There's also a bunch of work out of both Google and Levin's lab about stain-free tissue reconstruction and thicker slices using autofluorescence. I mean, one of the problems with life, of course, is we don't have to think of it at a time. So these sort of physical experiments are really important. But so I mean, I think that's the right way to do it. I mean, since then I've written some papers on 3D reconstruction. The thing is that mostly I found the sweet spot at the moment is finding the large patient populations with pre-existing data. And so there's scalability in many of those large patients. And then there's scalability in the lots of data from a single patient. And this multiplex stuff is lots of data from a single patient. But so I think that's a really important thing, but I don't think the right thing to do is just think about scalability. Like you're gonna every time you have case, you're gonna slice it into like 1,000 slices or 100 slices. I mean, so it's essentially a research thing once in a while. But if you develop the right technology, you should be able to get 3D image routine. So I think that's the cool thing to do, actually. But it's a great
I'm
question.
just curious because there's a lot of like spatial organization within three-dimensional.
No, yeah.
That question is that okay if I script it this way?
Sure, we're leaving a lot out. Yeah. I mean, you can impute. Well, the other thing you can do is billions of dollars going and I mean, as you know, there are all sorts of methods. There are all sorts of methods that take a few different views and then reconstruct the 3D scene. Maybe not perfectly, but pretty well. And obviously that's another thing to do is take a smart set. Like for instance, you take a section like this and sections like that and sections like that and try some of those algorithms. They're mostly much smaller images, but I've been very impressed by how good some of those algorithms seem to do. Yeah, I mean, I agree. I think there's a lot of signal. So, another thing, by the way, just in terms of new technology, I wasn't sure where to get into it, but so one of the things I'm really excited about involves self-supervised training, and in particular, methods that leverage the sort of detailed information that I was talking about here, in order to train a model to recognize information from ancient images. And so we have some nice results from one of our students. Basically, is doing self-supervised training for cellular versus non-cellular, and what seems to be when it's public, looks like it'll be published in the next place. More importantly, we got really a big delta in our ability to distinguish between pancreatic cancer and evil pieces. Using that as a lighting space representation versus. Any event, okay. So FDA. So and I know Elena is very deeply involved with the FDA. We've been working actually with a group doing essentially validation of pathology. So this is basically a package called a MVVT, which we don't know what it stands for, but it's basically a package involving data and analytic methods used to validate data. And so this is pretty well established in radiology, and that's got a race track or whatever that we developed for this. It's not as well developed in pathology, so at least as far as you can see, I hope it's looking for the FDA guide. This is kind of the FDA's first time of doing this, and they're using tumor infiltrating lymphocytes as their use case. So the idea is to essentially create a validation data set to be used by people who want to get their AI till algorithms and validate. And so there's a whole story there. The methodology here is very excessive as the FDA. That's a good thing. And so there are lots of pathologists who have been recruited to choose regions of interest, to label classified regions of interest. Then there's a set of statistical methods. So we've worked with them on that and actually contributed to do that on that home base. And then they look at the inter-observer variability of humans. Because think about it, if you have an AI algorithm that's going to emulate pathologists, then not all pathologists give you the same answer. And so there's a lot of observability. And so documenting the inter-human observability, but you have one pathologist or one radiologist and your test set is imitate that pathologist, and then the whole industrial world is dedicated to imitating that pathologist. What if that pathologist doesn't agree with other pathologists? I mean, it's a reasonable question. It's like, why is that pathologist so good? So there's sort of the whole issue of inter-observability, which seem particularly high in some of these pathology tasks. Obviously that exists in radiology as well. So data management and curation. So the whole process of data query, search, analysis, and the like becomes very important. Initially, when I got into the whole side of business, and actually I got into it by virtue of having worked with the computer vision foundry, Larry Davis. We got an NSF Grand Challenge Award involving analysis and land cover satellite. We got it, and then I did. So actually, then I went to Hopkins and started working with them, and basically had the idea that you can do the same thing with both side images, but with the time we're incredibly huge data. And so we managed to get IBM to buy us SP2 with a bunch of disks. Each disk was 4 gigabytes and like 200 disks. So and so basically, if you scan in the whole slide image without pressure, which we did very painfully for the dare, we have a robotic microscope and programs that look at different pictures and different type of you can put 2 or 3 of these things. So the thing was nuts. I mean, from a practical standpoint, the big grant out to optics and like I figured, I mean, I was an optics. I said, well, this isn't practical. I'm not going to say it's practical. It was like like $2,000-3,000 the whole slide image to store. I mean, this was expensive too. But in any event, so storing data is now much easier. But actually managing and analyze annotations, if you've got trillions of annotations, which have sort of a lot of spatial information you're trying to do with the text, fairly elaborate, is sort of an interesting thing. So Cancer Imaging Archive, which some of you guys. So we're funded by one of the grants, the Information Technology Cancer Grant that funds that. And so we do the pathology data for that. So this is a big deal if you're into this sort of thing. We've developed software that matches this kind of stuff. And one of our staff people, Eric Bremer, has been doing this wonderful job of essentially developing over curve based method for managing multi-scale computations. We actually got it over the data management business. The satellite yeah, which picked up its own citations. So it's back to that. The context there was clustering and clustering. That's a whole history. In any event, so the ability to, in a very natural way, lots of data computation is actually more of a problem than you think. The problem is not doing it. The problem is moving it rapidly because you want to be able to zoom in. So he's done a really good job with that. I'm trying to find the code and the paper from it, but it's going to be called a speaker too. And so we're using it. A few of our collaborators are. I'll skip over this and I'll end up by saying things about modeling human attention in that. So this is really a different part. Okay. So here we get rid of the Lego and we go back to good old school pathology class. So this is the scenario where we want to be able to better understand what sort of interobserver variable sort of classification a human is likely to give us. And the basic concept is that we want to observe what they look at and what power, and then build a deep learning model to simulate, essentially. So this is a cool project, I think. Greg Zielinski, unrelated to the other one, well, maybe he's on the same is our cognitive psychology department. And he's worked with Demetris Marks, who's the other beyond three of us, who's a computer vision guy. So they worked together for years doing eye tracking stuff. And usually not. And so we got together along with this wonderful grad student, Chuck Morty, and basically have developed a platform for tracking, continuing and capture what power like people have looked at. And then, so in this case, this is a prostate cancer. The request was give us the basic. And then if you actually look at the heat map of what people look at on the right, it's very similar to tumor segmentation. We actually got ground truth tumor segmentation. We also do it confident. These things we did it manually. So that's kind of a sanity check. But then what we're doing is essentially asking, can we predict the heat map, what people look at by modeling what the pathologist does, and then can we predict the individual trajectory of a pathologist and understanding that there's an intrinsic variability in what people look at. So we've gone through published a few conference papers on this. We've gone through our first set of experiments with 1,500 pathologists looking at 16 ECG. We're now about to go with 300 slides in a much larger stuff. We've got both funding and IRB approval pathologists, 100 bucks the crack place. So hopefully we're getting a lot of data. And then we're also looking for pathologists who will spend a lot of time as part of our research team, so we can develop part of our proposals to develop models to emulate the particular pathology. This is the task. So, pathologists. Essentially spend a lot of time with this thing. Then the idea is that you get enough data, you get a model that actually emulate what things specifically the person would look at. Which I think would be very cool. So it's kind of a basic sciencey thing, but the conjecture is that being able to evaluate pathologist behavior rather than saying we think we know the answer and we got the wrong answer could potentially be useful for getting much more nuanced decision support. We probably will check this decision support. Over there. I'm saying experimental. I mean, and there's a whole human factors component because people hate decisions, right? I mean, whether it's work or not, you know, it's really annoying. So then if you can be like really subtle about it, then the hope is that it doesn't have to do. The other problem is like, if your algorithm gets the wrong answer, it like freaks everything out. Somebody like the radiologist that about mammography and about CAD support, which has been around for years. They're both the good news is even though everybody says that data, it does want something to find things to get the same. The bad news is when you find something that you think is yes, usually you refer to anything. If you don't want to be on the wrong side of a lawyer who says, and even the algorithm pointed it out, doctor, is there a reason you didn't include it in the report? Right. So it clearly leads to bias towards over-reporting. And because basically false positives are quite likely to be reported. So a slightly lighter touch a bad idea if you want to prevent a lot of over treatment. In any event, so that's been a fun thing. So we are launching this big study. So okay, so wrapping up. One of the things I think that's most exciting, and this is something I was pushing before, the notion of foundation models that actually capture using semi-supervised, combination supervised, semi-supervised, that is, in some sense, the semantics of an area. So basically, if you were to go to a pathologist, take a snapshot of the slide and say, what is it? They would probably know. If you were to go to a pathologist and say, generate from nothing an image of a slide or a patch with a particular set of verbal characteristics, they couldn't, but they could think. And I think that the appropriate model would have a model to actually generate synthetic images. I mean, there's a lot of type stuff out now, but basically sort of much more like clips, like the photo studio, like Van Gogh, they basically describe something nuanced, they could generate it. But then more importantly, basically, like I showed you these bird cells in the butterfly together, and people who know a lot about life, about tissue, know that's crips and ducts and whatever, so basically the model said if you take out this certain area, it should be able to go. Anyway, bottom line is the conjecture is that if you reach the foundation model of a large, essentially a large image model, train it with people data, then you should be able to at least use its latent space and customize it to do things. So I cited this admittedly skewed evidence about who's attempt to do that, doing better from pancreatic classification. It's extremely weak evidence that the public, but I just say verbally that it's probably going in the right direction. So I think that's a really probably very important sort of scenario. Another scenario similar to that is there's the long tail of distribution. So having said that we're going to inventories and Lego, ordinary pathology is not going to be simple. And supervised methods, where you're going to get, if you look at almost any tumor type, there are going to be dozens to hundreds of subclassifications that pathologists are paid to look for that you don't have a lot of training. But it's not good enough to say, okay, there are 5 types of gliomas we're going to train for those, because like behavior pathology is not good because it should be down, but it's like, is it something weird and obscure? We really don't. And so the long tail stuff, we need a lot of nuance, and you need something much more like a large language model where you can actually sort of give examples and looking for this. So I think that the long tail problem is a really important problem, which is related to but a little different than the learning tissue problem. Anyway, so I think that's about it. I just want to give a shout out to medical imaging and computer science faculty. Most of these folks actually publish non stuff, computer science conferences, but a lot of focus is on medical. And we've got multi-institution, multidisciplinary teams for various projects with a lot of people involved, and it's a subset of some of the funding that the group does. And with that, I hope I saw everybody. So thanks. I'm happy to questions.